So hello and welcome to our second lecture on the biochemical basis of cancer, excuse me, where today we'll be looking at how cancer cells achieve immortality. To start with though, we're going to look at signaling pathway deregulation, typically pathways involved in promoting cell proliferation and how deregulation of that causes excessive cell growth. So the first pathway we're looking at involves KRAS. So RAS, there's different forms. So there's KRAS, NRAS, and HRAS. And they're a small family then of proteins, but they are all monomeric G proteins and important components of growth factor signaling pathways. The three of them have a high degree of sequence homology, but may have slightly different roles. But as I say, today we're concentrating on KRAS. So the signaling pathway that KRAS is involved in is shown here on the right-hand side of the slide. It starts with a growth factor binding to its receptor in the plasma membrane. So this is a dimeric receptor, the growth factor binds, and as you're familiar with, there's a conformational change that's propagated to the cytoplasmic domain of the membrane, which actually involves or contains a tyrosine kinase domain, and you get autophosphorylation of tyrosine residues in this tyrosine kinase domain following activation by the growth factor. So this phosphorylation of the tyrosines enables an adapter protein known as GRB2-SEM5 to bind to the phosphorylated tyrosine via its SH2 domain, and then via two SH3 domains. It combined to SOS, and SOS basically promotes the displacement of GDP from the RAS protein to the active GTP form. So note that RAS is anchored to the plasma membrane by this isoprenyl group. So this SOS is actually, I've got it down here for you, it's a guanine nucleotide exchange factor. So as I say, it stimulates the exchange of GDP with GTP to give the active G protein. And this process can be reversed by these GAPs. GAP is a GTPase activating protein that increases the inherent ability of RAS to slowly, under no other circumstances, to hydrolyze the GTP back to GDP to give the inactive form of RAS. So anyway, so RAS has been activated and has a number of ways of acting, but we're just concentrating here on this central pathway, which results in increased cell division. So RAS interacts with RAF to activate RAF, which is a serine threonine protein kinase, which in turn phosphorylates MEK, and then phosphorylated MEK is active and phosphorylates MAP kinase to activate the MAP kinase, and then the MAP kinase, either directly or sometimes via other kinases, can phosphorylate a number of important transcription factors, activating those transcription factors to bring about the expression of genes whose proteins are involved in promoting cell division. So these transcription factors, there's three shown here. There's MYC, which we're going to be talking about later in the lecture. Phosphorylated form is MYC is active. You've got phos and phosphorylated Jun, again, regulating the expression of genes that promote cell division. So we're going to talk a little bit more about RAS then, seeing it has this important role in the signaling of cell proliferation by cell division. So we're going to just again look at how KRAS is activated, and then we're also going to look at some activating mutations. So the RAS family of G proteins is activated when GTP displaces GDP in response to binding of the receptor tyrosine kinase. That was the receptor we just saw on the previous slide. GRB2 SOS complex, and this is generally it's called a GEF, which equals a guanine nucleotide exchange factor. So RAS is inactivated when the bound GTP is hydrolyzed to GDP by the inherent hydrolase activity of RAS, which is stimulated by these gaps. In other words, GTPase activating proteins, which SOS is an example. So here's the inactive RAS with GDP bound by the GF. The GDP is replaced by GTP. So it's not a phosphorylation of GDP, it's replacement of the GDP with the GTP to give the active RAS, and then these gap proteins can hydrolyze the GTP, or at least stimulate the ability of RAS to hydrolyze the GTP, returning it to the inactive form. So the active form of RAS can then interact with various effectors. They can be crosstalk, but will ultimately bring about a cellular response. So a common mutation in KRAS in human cancers is a point mutation to codon 12. So codon 12 is normally a glycine, but here there's a mutation to give aspartate or aspartic acid. And of course, the amino acids, glycine only has a hydrogen atom as its side chain or R group, whereas aspartic acid has a charged carboxyl group. So they're very different amino acids. And this results in the formation of hydrogen bonds with glutamine 61 and tyrosine 32 that do not occur in the wild type enzyme. And this brings about the loss of binding of the water molecule that actually hydrolyzes the GTP back to GDP. So hence the KRAS GTPase activity is lost and the KRAS becomes permanently activated. So you can see this in the little diagram on the right. So here's now aspartic acid in position 12. So normally it would be a glycine, but now we have the aspartic acid at position 12, close to the GTP, and it has a charged carboxyl group, which is this group here. So this charged carboxyl group, unlike the glycine, can form a hydrogen bond with tyrosine 32 down here, and that displaces the position of the tyrosine 32. So here you can see the position in the wild type enzyme, but because of this hydrogen bond, it's shifted quite a bit, so that hydrogen bond can form with the mutation, and also this aspartic acid group can hydrogen bond with glutamine 61. And again, you can see that's the position in the wild type enzyme. So the position of that glutamine 61 has also changed. There's a water molecule involved in here as well. But basically, this movement or change in the active site prevents a water molecule from getting in to be in close proximity to the phosphoryl group, the gamma phosphoryl group, in other words, the terminal phosphate of the GTP. So you can't get water in there, so you don't have the water available to hydrolyze that phosphoester bond, converting the GTP to GDP. So again, the net result is that KRAS becomes permanently activated. So let's move on and talk about P53, very important protein in cancer. So it's actually a transcription factor and it acts as a tumor suppressor. So if we just briefly look at the mechanism of action of P53, so it's a transcription factor and it stimulates the transcription of the gene for this CYP protein, an example of which is P21. And this CYP protein is an inhibitor of the CDK cycling complex. So the CYP, or P21 as an example, binds to the CDK cycling complex, and that inhibits this, this complex is inhibited. There's no longer the CDK activity. So the kinase activity is inhibited by, for example, P21, and that causes arrest in G1 and G2 phases of the cell cycle. So also note, and we're going to talk about this in the third lecture of this series, P53 is actually stabilized by DNA damage. So DNA damage results in activation of a kinase that phosphorylates P53, which increases the stability of P53, so that P453 is more active after DNA is damaged, arresting in G1 and G2 so that the cells have a chance of repairing that DNA before progressing. So here anyway, P53 inhibits cyclin-dependent kinase, the CDK, through increased transcription of the P21 gene. So let's look at some mutations that cause P453 to lose function. So of course with P53, because it's a tumor suppressor gene, it's loss of function mutations that are the problem in letting cells have unregulated growth. So we have basically the primary sequence of the P53 protein here with the different domains shown. So what's obviously important here is the DNA binding domain, which is a large segment of the protein shown in pink, but other domains, you've got the tetramerization domain, where it can form obviously a tetramer. You've got a negative regulatory domain at the carboxyl terminal, and at the N terminus you've got a transactivation domain, and after that a proline-rich domain. But you can see, looking at the DNA binding domain, you can get many mutations in this area, or this part of the protein, some with very high frequency, which we'll talk about in a moment. But basically, mutations in the DNA binding domain inhibit DNA binding and reduce its specificity, and therefore they reduce the effectiveness of P53 as a transcription factor to arrest cell cycle, induce DNA repair, and induce apoptosis. So you can see some hotspots here where mutation is very common, particularly here at 248 and 273. So let's look at those in terms of the structure of P53. So here's P53 interacting with the DNA in the promoter region of the genes it controls. So the DNA here is in grey. And if you look at the position of these residues, so it's normally an arginine, so which is R248. And of course, that's an amino acid with a positive charge. And that positive charge can interact with a negative charge on the phosphates. And similarly here at R273, positively charged residue interacting with the negatively charged phosphates in the backbone of the DNA. And those residues are important for strong binding of P453 to its target DNA molecule. So you can see that mutations to those residues that take away that positive charge are going to interfere with the ability of the P53 to bind to its target proteins. Another mutation that occurs with high frequency is to R175, but this is too far to be involved in direct binding to the DNA. Rather, it's involved with binding to co-activators, other molecules that are necessary for P53 to act as a transcription factor. So let's look at other common oncogenes and mechanisms of transformation. So we're going to initially look at c-Myc. So c-Myc has many important functions in a cell. So it stimulates cell cycle progression. It stimulates cell growth and division. It also stimulates metabolism. And remember that rapidly growing cells need lots of energy, so it can stimulate energy production. It also stimulates telomerase activity, where telomerase is an enzyme that maintains the length of the ends of the chromosome, which is very important for giving cells the ability to undergo cell division. And we're actually going to talk about that more at the end of the lecture. But Myc also is involved in regulating cell adhesion and motility. It promotes angiogenesis, and it also opposes differentiation. So differentiation if cells differentiate, then they're not proliferating. So that sort of also promotes unlimited growth of the cells if you're blocking their differentiation. So you can, based on this, you can see some of the problems you're going to get from a deregulation of MYC expression. So it can cause genome instability. It can cause uncontrolled cell proliferation. It can also cause escape from immune surveillance, so that the not removed by the immune system. It can cause immortalization, which is again via this stimulating or promoting telomerase activity. And as I also said, we'll talk about that a bit more later. And it can also cause the cells to grow independent of growth factors. And that's because MYC is downstream of the growth factors, so that you're stimulating the pathways promoting cellular growth downstream of the growth factors, so the growth factors are no longer required to stimulate the growth of the cells. So c-MYC dysregulation is common in many cancers. So we've got the frequency here of c-MYC deregulation in various cancers. So you can see in Burkitt's lymphoma, it's always deregulated. But in other cancers like ovarian, it's about 40%, prostate cancer about 70%, hepatocellular cancer, in other words, liver cancer, about 33%, melanoma of the skin, around 61%, and colon cancer, around 67%. So again, in many cancers, we get this deregulation of c-MYC. So let's see how this can occur. Oh, actually, before I go on, I should have also mentioned, I deliberately didn't mention it before, apoptosis, because this is sort of counter to what you'd expect for MYC. So it can also promote apoptosis, which is sort of an effect against cell growth, whereas the other effects are promoting cell growth. We're going to talk about that a little more in the last lecture of this series. So c-MYC controls cell cycle progression through G1S of the cell cycle. And it does this by, excuse me, regulating the activity of this complex cyclin E, CDK2, which is active in its phosphorylated form and stimulates or allows cells to progress through this G1S phase. So let's see what exactly MYC is doing. So, sorry, excuse me. So c-MYC again acts as a transcription factor along with another transcription factor known as MAX. So you get this heterodimer formed between MYC and MAX, which binds to a conserved region in the promoter of the cyclin D2 and CDK4, that cyclin-dependent kinase 4 genes. So you get increased transcription of these two genes, so you get more of these two proteins produced, which bind to each other to give this complex, and that cyclin D2 CDK4 complex binds this inhibitor protein, KIP1. And via some other proteins, you don't need to worry about these, but these are also upregulated by MYC, so they're target genes for MYC. They cause the ubiquitination of this KIP1 inhibitor protein, causing its proteasomal degradation. So KIP1 would otherwise bind to cyclin E CDK2 complex and act as an inhibitor of this complex. So the complex is not active in the presence of KIP1. But when you're expressing, or the cells are expressing lots of cyclin D2 and CDK4, then the KIP1 is now binding to the CDK4 cyclin D2 complex and being targeted for degradation, so that you don't have the KIP1 protein inhibiting the cyclin E CDK2 complex, enabling the complex to be phosphorylated by this CAK. I'm not a great lover of acronyms, so I like to know the full name, so I'll give it to you. So it's a cyclin-dependent activating kinase that phosphorylates the CDK2, promoting or giving rise to the active complex, are required to get the cells to progress through this G1S phase of the cell cycle. So MYC does something else as well, again, acting as a transcription factor along with MAX and another transcription factor, MIS1. It negatively regulates or downregulates the transcription of genes encoding cyclin-dependent kinase inhibitors. So these are other inhibitors besides this one. So you have less cyclin-dependent kinase inhibitors present, as well as more of the active cyclin E CDK2 complex present. So anyway, the net result is progression through G1S. So that, and you can see that then deregulation of c-MYC is going to cause deregulation of cell cycle progression, which is in this slide. So we can look at the mechanisms of deregulation of c-MYC. So one thing that can happen is you can get upregulation of the expression of normal c-MYC. And there's three ways that this can occur. The first one is insertion of a viral promoter upstream of the c-MYC gene, so that the expression of the c-MYC gene is now controlled by the viral promoter, and you can get increased expression. This can similarly occur with a gene translocation, where the c-MYC gene can move to a different chromosome, for example, and become under the control of a different promoter. And again, by being controlled by a different promoter, you can get excessive transcription. And you can also have a gene amplification, where you get multiple copies of the c-MYC gene, and having multiple copies just enables the transcripts to be made more rapidly. So you can also have mutations of c-MYC leading to upregulation of its activity. So these are, again, basically gain of function mutations that upregulate its activity. And you can also have upregulation of phosphorylation and activation of MYC by upstream oncogenes in signaling pathways, such as what we saw just a little while ago, RAS. So upregulation of RAS can upregulate the phosphorylation and activation of c-MYC. So let's look at some of these in a bit more detail. So here we're looking again at the first examples we saw on the previous slide, upregulation of expression of c-MYC. And really it's a similar information, but we've got some little diagrams now just to show you. So you can have the insertion of this retroviral promoter, or an enhancer for that matter, but promoter or enhancer upstream of MYC, so that MYC is now being regulated by the viral enhancer or promoter rather than its normal promoter, and that can cause increased expression of MYC. And the second example was the chromosomal translocation, and what could commonly happen is it can be translocated to the immunoglobulin gene and basically be controlled by the immunoglobulin gene promoter, which is expressed or controls expression at a high level, so you get increased expression of MYC. And you can also, as we saw, see gene amplification. So now we can see we've got 2 copies of MYC. So that by having the 2 copies, you get twice as much expression of MYC. So this middle one here, chromosomal translocation, we can look at that. And the example of this translocation is seen in Burkitt's lymphoma, where c-MYC is placed adjacent to the gene coding for the constant region of the heavy chain of immunoglobulin. So it's written as CH, the heavy region of the chain. And the expression of c-MYC is then controlled by the expression of CH, which results in upregulation of the expression of c-MYC. So if you're looking at the chromosome, c-MYC is at the ends of chromosome 8, whereas the immunoglobulin gene, you've got the variable and the constant regions near the end of chromosome 14, and simply a translocation or a swapping over, you now have the variable region of the immunoglobulin gene on chromosome 8, and you end up with the c-MYC gene at the end of chromosome 4, where its expression is regulated by the promoter for the heavy chain of the immunoglobulin gene, and the immunoglobulin gene, again, is normally expressed at high level, so you get high level expression of c-MYC. So now let's look at the upregulation of c-MYC phosphorylation and its activation. We're going to I'll just describe it, and then we're going to look at it with a diagram. So activation of RAS upstream of MYC results in activation of MAP kinase, which phosphorylates c-MYC at serine 62 to activate it. An activation of RAS also results in inactivation of GSK3, which is glycogen synthase kinase 3, through a separate signaling pathway. And GSK3, when active, phosphorylates c-MYC at T58, which stimulates its degradation, which I think is the next sentence anyway. But this phosphorylation of T58 promotes dephosphorylation at serine 62, and in activation of c-MYC and ubiquitination of c-MYC, leading to its degradation in the proteasome. So, but in this pathway, you get activation of RAS, causing inhibition of c-MYC degradation. So you get oncogenic activation of RAS not only results in activation of c-MYC through MAP kinase pathway, but also inhibition of its deactivation and degradation via downregulation of the GSK3. So a common site of oncogenic mutation of c-MYC is at threonine 58 or T58, which again results in inhibition of deactivation and degradation of c-MYC. So again, perhaps easier to understand in terms of a diagram rather than just the printed words or spoken words. So again, looking at the same thing, upregulation of c-MYC phosphorylation and activation. So RAS via two pathways can affect the level of active MYC. So overexpression of RAS, for example, which we saw earlier in the lecture, you'll get activation of RAF, which activates or phosphorylates MEK, which is a MAP kinase, which phosphorylates MAP kinase, and then the MAP kinase phosphorylates c-MYC here at serine 62. So you've got the phosphorylated serine 62, and that converts c-MYC to the active form to carry out its function. So active c-MYC is inactivated by a GSK3 dependent phosphorylation at T58. And that phosphorylation at T58 enables this PIN1 enzyme to bind to the MYC, and this P1 enzyme is a proline isomerase, so it actually causes a structural change in the MYC protein, which enables then this phosphatase PP2A down there, phosphatase, to remove the phosphate from serine 62, which inactivates c-MYC and also targets it for degradation. So the FBW7, which is an E3 ligase, which basically catalyzes polyubiquitination of the MYC, is active. So the ubiquitinated MYC is then targeted to the proteasome for degradation. So RAS, via this second pathway involving PI3 kinase and AKT, is downregulating this GSK3. So by downregulating GSK3, you're basically downregulating this inactivation pathway, so leaving more MYC in the active form activated by this other pathway involving the MAP kinase. So phosphorylation at T58 by GSK3 targets c-Myc for degradation. So consequently, as you'd expect, mutations to this residue T58 can cause the loss of inactivation of c-Myc. Okay, so let's go on now and talk about mutations to other important genes involved in the regulation of progression to cancer. And these are the general caretaker genes. So first of all, there are a group of DNA polymerases used in DNA repair, which you should have come across before. So they repair DNA following DNA damage. But a subgroup of these repair enzymes repair DNA containing chemical modifications of bases and missing bases. And these are called lesion bypass DNA polymerases. So DNA polymerase beta is an example of a lesion bypass polymerase. And about 30% of human tumors show mutated forms of this enzyme. And one mutation that seems to be reasonably well understood, the mutant has reduced fidelity, or that means, in other words, accuracy of replication of DNA, resulting in more errors. So the polymerase, rather than repairing the DNA, actually can increase the number of errors in the DNA, of course, obviously promoting the transition to cancer. There's also the BRCA1 and BRCA2 proteins that are also involved in DNA repair. So the repair enzymes that repair double-stranded breaks and also recruit other DNA repair proteins to lesion sites where there's damaged DNA. And here you can get a loss of function mutations which lead to accumulation of lesions, and these are associated with breast and ovarian cancers. So the last major topic for this lecture is the telomeres. So telomeres, hopefully you've heard of them before, but telomeres are a repeat sequence of bases, and the sequence is actually TTAGGC, but you don't need to remember that. And this sequence is repeated typically 3 kilobases up to about 20 kilobases of DNA length, and it protects the ends of the chromosomes. So remember that lagging strand synthesis of DNA replication leads to shortening of the telomeres in somatic cells. This limits the number of times they can divide, and the loss of telomeres triggers p53 activation of apoptosis, leading to apoptotic death of the cell. So once they lose their telomeres, they become senescent and undergo apoptosis. Embryonic stem cells and stem cells express this telomerase enzyme, which maintains telomere length, but somatic cells generally have no or low levels of telomerase expression. So somatic cells, as they divide, they lose their telomeres and ultimately become senescent. So I'm going to show at the end of the lecture a little video just showing this problem of lagging strand synthesis, how the lagging strand end gets shorter with each DNA replication, and how telomerase reverses this. But we'll do that last because I can't actually, I've got to open that as a separate share folder because I'm doing this from a PDF and it won't work from the PDF. So we'll talk a bit more about telomerase first before we look at that little movie. So that would be the movie. We'll see it in a moment. So here, just now considering telomerase and cancer cell immortalization. And this is one of the key events, again, that gives cancer cells their immortality, that they can replicate unlimited. And I think some of the first cell lines taken from cancer cells was back in the 1950s, and certainly in the test tube or in the lab, they're still proliferating just fine. They're basically immortal. So most cancer cells produce telomerase to avoid telomere shortening, which leads to apoptosis, or the shortening of the telomeres. So if we look at an embryonic stem cell, it typically has between 3 kilobases and 20 kilobases of the telomere repeat sequence, the TTAGGG. So N is a very big number here. And these cells can undergo indefinite replication, which of course is a characteristic you need in a stem cell. It can replace itself over and over. But in somatic cells, the telomere length is much shorter, and they have a limited number of replications. It varies depending on the type of cell, but a limited number of replications. So after, who knows, but something, maybe 100 cell divisions or something, the cell will become senescent because there'll be no basically telomere sequences left. And when there's no telomeres left, this basically stimulates apoptotic cell death. So the senescent cells mean basically the cells that are going to die by apoptosis. If you look at cancer cell, on the other hand, you see that there's the telomere length is very long. There can be up to about 55 kilobases of the repeat sequence, the telomere sequence. So that's even longer than in the embryonic or stem cells. So again, these cells can just keep growing and growing, or dividing and dividing. The telomeres don't get shorter because the telomerase is maintaining the length of the telomeres, and the cells, as I say, basically can go on dividing indefinitely. In other words, the cells become immortal. So what regulates the expression of telomerase? Well, it's perhaps not too surprising in terms of other things we've covered already in today's lecture. We have c-Myc, again upregulated in many cancers, as we saw, as a positive regulator of telomerase gene expression. So with unregulated c-Myc, you also get increased telomerase activity in the cell. And of course, the tumor suppressor factor p53 does the opposite thing. It represses the expression of telomerase so that inactivating mutations to p53 are also going to promote increased expression of the telomerase gene, maintaining the length of the telomeres that you see in cancer cells. So both c-Myc and p53 regulate telomerase expression. And again, this very long telomeres is consistent with immortality of cancer cells. So to finish off, I've got to open the little movie program. So I'll just stop the share on this one, and I'll finish off, and you can just see the movie. I'll also put the movie, or if I haven't, well, probably by the time you see this, I will have already done it, but I'll put it on LMS so that you can see the movie separately if you want to see how the telomerase maintains the length of telomeres. So new share is what I need.
The ends of linear chromosomes pose unique problems during DNA replication. Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template. Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template, the lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete. The final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed. The 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA. However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats, called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction and adds additional repeats as it moves down the parental strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.